Some children are born with eyes that never close to the light, pupils wide open, in darkness or in sun, never narrowing, never adjusting. Not a nerve injury, not a shadow over the eye. The muscle that should draw the pupil shut cannot contract. The actin it is built from is mutated. ACTA2. ACTA2 sits on chromosome 10. It encodes one protein, smooth muscle alpha actin. Smooth muscle lines the walls of your arteries, your gut, your bladder. Not the muscle that lifts your arm. That is a different actin entirely. This one works quietly, and it never stops. It links into long filaments, grips the motor protein myosin, and pulls. That pull gives the aorta, the great artery leaving the heart, its strength. Without it, the wall gives way. For decades, cardiologists watched families lose members the same way. A tear in the aorta, a dissection at 40, at 30, sometimes younger. In 2007, Dongchuan Guo and Diana Malevich at UT Houston traced it through family after family to a single gene, ACTA2. In about one inherited aortic case in 7, this gene carried the damage. One broken copy is enough. So why does one faulty copy cause disease? The inheritance is dominant. One mutant copy out of 2 is enough, and the healthy copy cannot make up the difference. The mutant actin is still built, and it fouls the filaments it joins. In smooth muscle cells from patients, the actin comes out disordered, unable to assemble cleanly. The wall loses tension, the tissue starts to splay, and the numbers are sobering. In a registry of 277 carriers, 3 in 4 suffer an aortic event in their lifetime. 1 in 4 of those kills at the first episode. Some tear below 5 centimeters, under the old surgical threshold. Start with what healthy actin does. In the arterial wall, single actin molecules link head to tail into long helical filaments. The filaments braid into a cable network running through each smooth muscle cell. Myosin grips the cables and pulls. The cell contracts. That contraction does 2 jobs. It sets blood pressure, narrowing or opening the vessel, and it holds the aortic wall taut under billions of heartbeats for a lifetime. Weaken that force, and the wall dilates instead of holding steady. The aorta swells like a balloon with every beat, until one day, the pressure opens a tear. But one position stands apart: arginine 179. Most variants fail one way: too little contraction and the aorta dilates. This arginine does something stranger. It still breaks the filaments in the cytoplasm, but it also slips into the nucleus. Because this actin is not only a building block, it is a gene regulator. There, it helps mature the cell into a committed contractile worker. The mutation cuts that supply in half, so the cells never fully grow up. And immature dividing cells crowd small vessels from the inside, narrowing them. One mutation, 2 opposite problems. The aorta, a large vessel, dilates toward a tear. The brain's small arteries narrow toward stroke. And the pupils? The iris dilator is smooth muscle too. It never matured. Those open eyes were never nerve damage. A window onto the aorta's fate. In the most severe form, called MSMDS, every smooth muscle organ is under threat. The ductus arteriosus, which should seal at birth, stays open. The bladder cannot empty. The gut slows. The brain's small arteries narrow. And by age 25, in every untreated patient, an aortic event has struck. Families raise these children listening for the next emergency, monitoring, imaging, deciding when to operate before the tissue decides for them. Reach the aorta before it tears, and the outcome is far better. That is what today's care is built to do. Today, no approved treatment changes the course of the disease, but a single-letter mutation points straight at what a cure might be. If R179H is one wrong nucleotide in every cell, what if you could correct that letter? That is the idea behind two programs building CRISPR base editors. A base editor is a molecular pencil that rewrites one DNA letter without breaking the double helix. In mice carrying the human mutation, a single dose extended median survival almost fourfold. Researchers are moving toward human trials. Nothing has entered a clinical study yet, but now the wrong letter is known, and a tool exists precise enough to correct it. 17 years. From families who kept losing members to a tear in the aorta, to a gene, to a mechanism, and now to a molecular pencil aimed at the right letter. The families tracked down the research. The families built the registry. The ACTA2 alliance keeps them connected to each other and to the science. Found in the arterial wall, carried by families. Find them at acta2alliance.org, the Gene Channel.